So the first thing, so the block 1 here is sort of like a go at your own pace sort of deal. The first part of it is that I'm going to give you an opportunity to go through your notes and complete the summary sheet. The summary sheet is actually at the bottom of day 4. But it's this sheet right here, which is the characteristics of the 6 kingdoms. So this is a self-review of the unit. Hopefully, as you go through here, a lot of this stuff will be familiar to you. So you go through and say, okay, archaea and bacteria, are they prokaryotes or eukaryotes? Hopefully at this point you remember those are prokaryotic. They are prokaryotes because they do not have membrane-bound organelles, they have no nucleus, etc. Are they multi or unicellular? Well, again, hopefully at this point you know that archaea and bacteria, prokaryotes, can only be unicellular. So I'm gonna get you to go through this. It's an excellent review, and to just make that on your own. It's also a great reference to have for the quiz. If you really don't want to make it, I mean, I guess I can't force you. I gave you a completed copy because I also gave you a completed copy just in case. Oh no, this isn't right. I also gave you a completed copy. Wait. Why is it not showing the bottom here? There we go. So it says there's a completed version here at the bottom. I guess you can just download it if you want. I don't recommend it. I gave you the completed copy so you could check your answers and just double check to make sure that you're clear on the properties of all these. But I'm getting to it. Jaleel. So the quizzes are always written during block 3 of whatever, the last day of whatever unit. So today is the last day. Today's day 5 of diversity. So the quiz is now available. I recommend that you write it during block 3. In order to access the quizzes, you just go to the quizzes tab at the top, and you should see it listed there. It says current quizzes, diversity unit, summative quiz. Okay, when you click on it, it should tell you what the time limit is, but it's fairly lengthy. I think it also tells you the number of questions, and it's open book. So you're meant to write it during learning block 3. I ask that you write it before tomorrow at midnight. So if you really do feel like you're unprepared to write it during learning block 3 today, you'd like to go back over something, you have some questions, that's totally fine. Please feel free to resolve that first and then write your quiz. But if you're not going to complete it before tomorrow at midnight, please let me know. We need to then discuss what your extended deadline is going to be. It's a species spotlight assignment poster, a poster of Google Slides. It is a Google Slides presentation. I used to do it as a poster, like an actual physical poster, but obviously you have no way to hand in a poster. And it's just easier to do it as a Google Slides presentation. So it doesn't have to be Google Slides. You could potentially use another program to make the slideshow, but Google Slides is easy. So I recommend Google Slides, but if you do want to use something else, you're welcome to do that. For some reason, when you hand in a Google Slides in Brightspace, it automatically converts it into a PowerPoint presentation, Microsoft PowerPoint presentation, and then it saves it as a PDF. So what a convoluted way to hand in a Google Slides presentation, but that's how the API works for this. So it converts it into a PowerPoint and then saves it as a PDF so that I can comment on it. So if you wanted to do it in PowerPoint, if you have PowerPoint, you're welcome to do that as well. I guess you're skipping a step, but you don't have to. Google Slides is perfectly fine. Will the summative quiz be approximately the same length as a practice quiz? No, not at all. It's much longer. I think, let me double check this. I mean, keep in mind, it's questions that are encapsulating the entire unit. So it's not going to be like one of the practice quizzes. It's significantly longer. Now, are the questions similar? in scope to the practice quiz. Yeah, they're sort of similar in scope. There's some challenge questions. Keep in mind it's open book. So, and also I should mention that if you're at home and you really have a question during learning block 3 while you're writing the quiz, you can just jump into the Google Meet and just ask. I'll be here. I mean, ideally, that's why I ask you to write it during learning block 3, is that there's support available if you have a question about one of the questions on the quiz. If you're writing it later outside of class time, and well, I'll do my best to get back to you, but no guarantees. But if you are unsure about a particular question, like let's say you think a question doesn't make sense or there's a problem with the question, I think I've figured out all the bugs at this point. But if something doesn't seem right, please make sure that you actually send me the full text of the question if possible, because I'm not able to, if you just say like number 5, it randomizes the questions. So your number 5 and my number 5 could be are definitely different. So there's really no way for me to know which question you're talking about. So try and be as specific as possible if you're going to email me, but a question from the quiz. Don't just say like it's question 2 or something. Let me just look at the number of questions here. I can just tell you the answer to this. There are 31 questions, and the time limit is 90 minutes. That's 3 minutes per question for a multiple choice / choose all that apply. Watch out that some of the questions are choose all that apply. I mean, I think I make them clear in the question, but you do need to pick all the ones that are correct in those ones in order to get the mark. But yeah, 3 minutes per question. That's a lot. Like that leaves you time to like look stuff up while you're writing. So, and again, it's designed to be open book. So take advantage of that. For the species spotlight assignment, do we just submit the slides or do we record ourselves? So for this particular assignment, the species spotlight assignment, you only submit your slideshow, your completed slideshow. You don't need to submit an actual presentation of the slideshow. After this point, all of the summative assignments are in some way presented. So the one for the next unit, and we'll get more into the requirements for that tomorrow. But that one, you're going to be designing a presentation or some type of video that is going to explain the process of meiosis, which we haven't covered yet, so you don't know what that is probably. But it is some type of video. And the same goes for all of the subsequent summative assignments. They are research assignments where you have to present them in some manner, probably using a tool like Loom or something like that. It doesn't have to be Loom. So they are and part of the reasoning behind that is that even in the hybrid system, I basically never talk to you or get to hear your voice or like see what your understanding is as you communicate it. And keep in mind that you're only communicating with me, so like no one else sees these presentations. But it's a way for me to get to know you better and actually like assess, are you able to communicate about these topics? Because otherwise I really have no idea. In some quads this year, I haven't even spoken to some students ever. So that's that's a little weird. That usually never happens. So anyway, it's a way for me to have some type of actual personal contact with you. Where do you find the Kingdom practice sheet? Again, it is on, I'll go back to that. I believe it's at the bottom of day 4. I can double check. Yeah, there it is. So at the bottom of day 4, it says consider completing this summary sheet. You just click on the link there for this summary sheet, and there it is. Is it 6 slides, including the title and the citations? Are you talking about for the species spotlight assignment? I would assume. Let me double check that. Okay, you need a title slide, taxonomic levels for your species, visual life cycle, image with descriptions, dichotomous key, works cited. That's 6 slides. Yes. Keep in mind that it doesn't have to be exactly 6. If you want to take 2 slides to do your visual life cycle because you just ran out of room to describe it, well, take an extra slide. If you didn't have enough room to put the total table for the taxonomic levels, then just have it go over onto 2 slides. Like it's really not a big deal if there's more than 6. Yeah, absolutely, it can be longer than 6. Yeah, so 6 is just like the bare minimum. You must have at least 6 because you have to cover all of these topics. But absolutely, and that goes for all the presentations, by the way. They have sort of like a general guideline in terms of slides, but if you need to spread information out onto multiple slides, I recommend that you do that to make them easier to read. So definitely you can always put more slides. It's not a problem. How detailed does it have to be? Again, I'm assuming, Kim, that you're talking about the species spotlight assignment. How detailed? It needs to include all of the required information. So like it discusses that in the instructions, like what you need for each section. Perhaps that's a better question that I can answer one-on-one, because I'm not sure what about that you're asking. If you have a start to something and you're wondering, is this enough? Like, have I described this in enough detail? Why don't you just email me and send me what you have and tell me what to look at specifically, and I will just tell you if that is in depth enough. But we did go through it together yesterday. Do you switch over classes tomorrow? No, I don't think so. No, that doesn't sound right. I think we have 10 days together. We would be completing 2. Oh, because normally you would switch cohorts, right? No. So really the cohorts don't mean anything right now, Sammy. They you're in this for the entire first block, which I think is 10 days. So normally it's cohort A would come home and cohort B would go to school or whatever. But we're not doing that from home because everybody's here. That's correct. Yep. On May 6th would be 10 days. Yeah. So course 4 starts on May 6th. We're only done the first unit. We're going to fit in 2 units before the switch over to your other course. So Kim, I'm not sure if I answered your question regarding length here or details. Maybe email me specifically if you're wondering about is something enough stuff. I would say it's better to be more thorough than less thorough for all of these things. Are you describing your life cycle in enough detail? Well, do you have an image for your species for the different parts of its life cycle? Have you numbered them? Have you described each of those steps and what happens in each of those steps of the life cycle? I mean, like, as long as you're including the required information, you should be good. For your dichotomous key, did you choose 5 other species, so you have 6 total, including your own? Did you show pictures of all of them with their scientific names? Did you create a dichotomous key with good dichotomies that clearly identify all 6 of them? Then you're good. Then that key is functional, and you've got enough for the dichotomous key. I mean, like, you just have to make sure that you have all the required information. I was going to get to the assignment, the vocabulary list in a second, but I can answer your question, Kim. Does the vocabulary list have to be grammatically correct? Yes. You're asking a grammar question. Does the grammar that you use in your description of the word have to make sense and be understandable by me? Yes, it does. I'm not going through and correcting your spelling technically, but keep in mind that, like in any assignment like this, yeah, you should make sure that things are spelled correctly. And you should go through and make sure that your grammar and syntax, the way that you've arranged sentences, is sensical and actually describes your species correctly. I mean, grammar is important for good communication. So yes, the short answer to that is you do have to have grammatically correct definitions. Do they have to be dictionary definitions? I'm not sure if that's what you're asking. They don't have to be dictionary definitions. They are in your own words, the way you would describe a specific term to your friend, or you would describe it to your parent. It doesn't necessarily have to be the dictionary definition. It should not be the dictionary definition. However, I'm not asking you to intentionally not use scientific language. If you understand the vocabulary from the unit and you're using that vocabulary as part of your explanation, that's fine. And it's also helpful, by the way, to do that. Trying to incorporate the new vocabulary that you've learned in order to describe things is useful. So try and use your own breadth of scientific knowledge to describe these things. But it shouldn't be a dictionary definition. Okay, it may be similar to the dictionary definition. You're describing in your own words and you have relatively mature scientific language that you use. That's fine. That's totally fine. I'm not saying dumb it down or anything. Like you use whatever language you feel is appropriate for you, but they should be grammatically correct and understandable. Not sure about the way you reworded. If you're not sure about something specific, like you have a specific example, again, also email me. Just give me an example of what you're talking about, and I'll let you know if it's acceptable. Does a vocabulary list have to be a table, or can it just be 30 terms? Please use a table. So it asks for a table in the instructions. Number your words. Okay, so term 1, term 2, just like put numbers in 1, 2, 3, 4, 5, 6 up to 30. That's also helpful for you to number the rows in the table just to make sure you actually have 30. So I highly recommend that you number them. It's also easier for me to get through the count because I occasionally miscount, and you need to have 30, a minimum. Ideally, guys, by the way, you're just adding all the new terminology that you find and having a minimum of 30 in there, not I've got 30, so I'm going to stop now. That's not exactly how the assignment is meant to work. It's meant to be a compendium of new vocabulary from the entire unit, but you have to include at least 30 words. So it shouldn't be necessarily ending at 30 for most of these units. But please use a table. I used a bunch of synonyms, so I'm not sure if it's the right scientific language. That's fine. If you're using common language synonyms for things, as long as they are true synonyms and they actually mean the same thing, then that should be fine. Printing something down here. But the overall meaning, the correct meaning of the term has to be maintained. So if by using a synonym or maybe a euphemism, you have bypassed the actual meaning of the term, or sort of misinterpreted the meaning of the term by using a synonym or perhaps a euphemism, then that's not good. So it does have to maintain the correct actual meaning for the word. So hopefully that answers your question. Can you go over the part of the species assignment about the taxonomic levels? Yes, I can. I can pull up. Give me 1 second. Actually, let's do it this way. So this isn't perfect, but essentially what you're doing is you're having one column that is domain, kingdom, phylum, class, order, family, genus, species. Then a column for your species. So which domain is it in? Which kingdom is it in? Which phylum is it in? Which class is it in? And then a third column, not shown here, okay? A third column on the right that is a explanation of why the species fits into any particular grouping. So for example, if it's domain eukarya, what makes something a eukaryote? Why does your species fit into domain eukarya? What about it makes it a eukaryote? And just explain that. It has membrane-bound organelles. And actually, that's all really you need to say if it's a eukaryote. It has membrane-bound organelles, including mitochondria, nucleus, etc. Okay? That makes it a eukaryote. Kingdom Plantae. What makes this particular organism a plant? And then just explain, like, what is the thing that makes something a plant that all plants have that your thing has? That's why it fits into that level of taxonomy, into that grouping, that taxa, or that. Hopefully that explained it, Jaleel. So that's the table that you're making, okay? And it goes all the way down to species. There is a reason, obviously, that it has been categorized at each level, and then you just need to tell me what that is. And then, yeah, it requires research. That's the point of the project. Okay, any other questions about the assignments? Okay, so just to discuss due dates really quick. Today, you should be done your vocabulary list. Because you've been... Oh, sorry. Okay, Jill, give me 1 sec, guys. And also, do you have to actually draw the biological drawing? No, you do not need to draw it. You can absolutely get that off of the internet. For this project, it's fine to use one from the internet. I think I mentioned that in the instructions, but you're welcome to use one from the internet for the image. Is that example posted? Yes, that is posted in. If you click on the assignment, it's one of the things that's attached to the assignment. It's set up sort of in a poster format because this previously was a poster, but each one of the little areas on the poster is its own slide, and it's not detailed. Obviously, it's not a complete dichotomous key or a complete table, but it's just to give you the general idea of what you're supposed to do for each thing. Okay, it's also outlined if you click on the assignment. It goes through, and there are headings for each section explaining what goes in each section. And you can also look at the rubric. The rubric explains what I'm looking for each section, and it just mentions it. So, in fact, I highly recommend that you do look at the rubric because that's what I'm going to use to mark it. Yes, they do. Interesting, Kim. So you can do that. So for example, if you have a bunch of related terms, so you've said like these terms are all, domains, and then there are these three, and then this is what each of those three terms means. Yes, each one of those like sort of subcategories I would count as a separate term. Yep, that's fine. You can count that as part of your 30. Yeah, some people organize it that way. I would say that's more difficult for me to mark, but if that's easier for you to organize in terms of making a good study note, like you feel like that is a better way to make it so that you can remember the terms and so that it's easier for you to study and to look things up in your own resource that you've created, then do that. Remember that the purpose of the vocabulary list is to be a study tool for you. It's to help you think about each term, decide what its meaning is, and then when you do that, when you think about the terms and explain their meaning in your own words, you're actually helping to commit them to memory. That's the purpose of the vocabulary list. In previous years, I have asked students to create that vocabulary list, but I didn't collect it as an assignment. It's meant to be a tool for you to help learn the complex vocabulary of the course, of which there is a great deal. And it's a mark booster. So it's a skill, basically, is to be able to do that, and something that you can use in other courses. Certainly in biology, you should pretty much always do it. But I'm marking it in this case because it kind of makes sense. It helps demonstrate your understanding of terminology, and it's an easy thing for me to mark, or it's relatively easy. But really, it's for you. And so if you're only doing 30 terms and cutting it off, or you're not really putting it into your own words, or again, you're shortchanging yourself. Like the purpose of it is to help you learn the content. So don't shortchange yourself. And if you need to slightly organize it slightly differently in order to be more effective for you as a study tool, that's fine. That's the purpose of it. Do you have to include why those terms are important as well? Yes. So that is part of it. Some of them are obvious from the term that this is related to diversity in this way. I mean, it's literally the word biodiversity or something like that. But some of the terminology that you're going to add is more tangentially connected to the idea of diversity and biodiversity. If it's more tangentially connected, you do explain, and you are asked to explain, how that term is connected to the idea of biodiversity. Why are we studying it in this unit? Why is that an important piece of terminology? So keep in mind that the terms that you're supposed to be selecting are meant to be the important terms from the unit, the core terminology. So, for example, so if you're going to pick, chondrichthyes or dinoflagellate as a word, okay? And those ones may not be on your list. They probably aren't on your list. Those terms are not core terminology from the unit. They are words that have come up in what we have discussed, but they're not really core terminology. They're not about diversity. They are a specific group in taxonomy or whatever. You can use some of those. Try not to make your list about those types of words. You can use some of those, but certainly if they're not obviously core terms about diversity and about, specific areas of diversity that we've used in the course, you do need to explain its connection to diversity. For example, if you're going to do something like eukaryote, well, eukaryote in itself, the idea of a eukaryotic organism is not necessarily obviously connected to the idea of diversity. But when you define it, you could also say it is one of the three main domains of life, and it is used to categorize a huge quantity of organisms, all of which share its characteristics, which you would discuss in your definition, that it has membrane-bound organelles. Anyway, you want to connect it back to why we are studying that particular thing in diversity. So and the function as well. Sorry, just to get to the end of what you mentioned there. So you're also going to talk about its function if it has a function. Do you do that for the subterms? Yes, you do. You do it for all the terms. That's the purpose. Okay. So, like I said, it's the last day. This is the day typically where everything comes due for the unit. As I mentioned, the vocabulary list is probably done. We've covered all of the content for the unit, so it's not like there's more content to cover. Although you might run into some additional terminology while you're researching your project, which we're going to work on during block 1 here. So if that's the case, then you would add it in, but you can hand it in anytime. I've noticed I've already got 3 or 4 of them in. Fantastic. There's no reason not to hand in your vocabulary list if it's done at this point. So the vocabulary list should be handed in today. There's no reason not to hand it in today. The project, I would respectfully request that you have it in by midnight tomorrow. So you can take, like I said, take the first block to complete it today, and then tomorrow is where I would ask you to hand it in by midnight. If you're not going to hand it in tomorrow by midnight, which is the official deadline, please just let me know. The idea behind letting me know is that we are going to negotiate on a reasonable extension, and which is totally fine if you need that. But we do have to negotiate on a reasonable extension because, and again, don't take this the wrong way, but students do have a tendency not to set a reasonable extension for themselves, and then they end up getting themselves into a lot of trouble. The purpose of us doing it together is to prevent you from getting yourself into trouble and saying, well, as long as I get it done by, May 6th when I'm moving over to my other course. Terrible idea, okay? What always happens when students do that is they switch over to the other course, there's a ton of work in their other course, and then this gets put on the back burner. They come back to this course again after having not done it, and then now they're really in trouble. So now they're weeks and weeks behind, and we're going to get right into the next thing. When you come back, you'll be working on other projects, other assignments, and it honestly will continuously pile. This has happened every single quad, and every student I've had that has not gotten the credit for this course has done that. That is the reason that they have not got the credit for this course. So please let me know if you're not going to get things in on time. You can propose when you email me what your alternative deadline is that is reasonable. And I'll probably just say, sounds good, that's reasonable. I'll see it then. But we do need to negotiate a reasonable extension on it. I do not want students to get into a bad way and not get the credit for this course. I hate that. That is like one of my, it hurts me in my heart that like students spend time and effort in this course. And then at the end, just because they just made some poor choices regarding that, they really hamstrung themselves and they were not able to get the credit at the end, which is terrible. So anyway, I don't want anyone to be in that situation. I really, really want everybody to get the credit. So do today, vocabulary list. Work on species spotlight assignment, complete by tomorrow at midnight. Work on quiz in Learning Block 3, ideally. You don't have to write it during Learning Block 3, but that's your time to get support for the quiz, and complete by midnight tomorrow. Okay, so those are the things that are coming due. Basically, Block 1 is your opportunity to do those things. Okay, not the quiz. The quiz is for Learning Block 3, but Block 1 now until the break is your time to work on your species spotlight assignment, and you can just hand in your vocabulary list. Maybe you need to add a couple more things to it, but that is what you're working on during Learning Block 1. When we come back for Learning Block 2, we are going to get right in to the second unit, which is genetics. And yeah, wow, it gets heavy real fast. We're going to talk about DNA transcription and translation. And we're going to do a little note together. We're going to talk about cell organelles, do a little review of the organelles of the cell. And then that's pretty much it for today. It's not a ton of stuff from the next unit, but we are going to get started. And I will discuss very briefly what the assignments are for the second unit, although it's similar to Unit 1. There's a major assignment, there's a vocabulary list, and there's a quiz. It's pretty much always the same. Okay, before I let you guys go, and remember that if you wish, and I highly recommend it, you should start by completing the summary sheet of the kingdoms. So start there. If you don't want to do that, you can look over the completed version. Again, it's up to you, but it certainly is in your best interest to complete it yourself to help prepare for the quiz. It's a great review because it also forces you to go back and look at your notes if you're unsure about something. Then you're going to complete the species spotlight assignment and your vocabulary list. Hopefully hand them in. And then I will see you during Block 2 at 11:05. Any questions? I will, of course, be available in the chat the entire time while you're working. Okay, I don't see any questions, but if they do appear, I'm happy to answer them. I may post it in the chat, or I'll pop back on here if it requires any kind of explanation. All right, guys, I will see you at 11:05. Good luck. Okay. If you're coming in late, no big deal, just let me know. Let's take a look at what we got going on for the second half here. So that's it for diversity. Although a lot of the concepts that we discussed in the diversity unit carry over to genetics and evolution and some of the other things we're going to look at. So it's not like we're never going to talk about any of those concepts again, but especially things like phylogeny and stuff will come up in evolution. But we're not going to dig into specific kingdoms, except for plants. Obviously, we're going to do a whole unit on plants. But what we're going to do now is we're going to get into genetics in a little bit more detail. So we're going to start with the very small in genetics. We're going to go down to the molecular level and talk about what are genes and what is genetics at the molecular level. Why do we care about it? How does genetics work at the molecular level? And there is a little bit of review from grade 10. In grade 10, you would have reviewed the cell organelles. And so you're going to do a little bit of that as well, a little review, because you need to know what the organelles are and what they do, because obviously in genetics, the organelles are doing something. They are participating in the process of expressing your genes. And so you kind of have to understand what the organelles are in order to figure out how they're playing a role in terms of how to convert DNA, genetic information, into a product, into an organism, into a physical structure. So I just realized you can't see this. Start by turning that on. All right, so we are going to start with a little note together where we're going to look at some basic cell biology stuff and DNA in a little bit more detail. So we're going to go over that together. There is a component when I'm done that, a component that you're going to do. You're going to read over page 1 and 2 of the course notes that are available right here. There's the note package, this note package. And that part is like a review of organelles and their function. There's a little coloring activity. You don't. It's very awkward to do in Canvas, so you can do the coloring activity if you want. You don't have to, but you should do the cell labeling. So then there's an opportunity to label the key structures. We'll take it up at the beginning of tomorrow's class. So we're gonna start with that. What are the structures in the cell? What do they do? That's a review from grade 10, so I'm going to kind of let you guys do that on your own, but ask questions, obviously, if necessary. Then there are some review questions on page 3. Again, that's review from grade 10. So it's some questions about the function of various organelles. Most of it's contained in the reading on page 1 and 2 of the course notes. When I say 1 and 2, I mean 1 and 2 of the course notes for this unit. So I separate them out that way. There is a little review video on organelles, which you should probably watch at the end. It goes over in a little bit more detail and excellent animations what each of these organelles are doing, and you do have to be familiar with them. So organelles, in general, what they look like in terms of being able to identify them and what they do is sort of a key part of understanding any type of cellular biology. And genetics is a component of that. It's the biochemistry component of cellular biology. So this definitely is key information that you need to know for this course and pretty much any future biology course that you'd ever take, unless you're really studying the macro like ecology or zoology or something like that. Then maybe this wouldn't be your bag. But pretty much any other area of biology, you're going to need to know about the biochemistry of the very small, the organelles. So there's that little video, and then there are these 2 videos at the bottom, which may be review for you, or they may actually be totally new information for you, depending on who you had in grade 10. So if you're in my class in grade 10, you may actually have watched one of these videos, but we did talk a little bit about how you go from DNA to a protein. So in that case, these videos won't be review for you from last year. If you didn't talk about it all, that's totally fine. We're really going to start at the very beginning and go over this process again. So both of these videos are about transcription and translation, or protein synthesis, it's also called. In other words, how do you go from having a DNA molecule to a protein? How do you actually build a protein from the instructions in a DNA molecule? Both of these videos look at different aspects of that translation process, or transcription and translation process. And we are going to spend a great deal of time tomorrow looking at those processes in detail. These 2 videos are to serve sort of as a refresher, if you've talked about them in grade 10, or a broad introduction with excellent animations, by the way, in terms of how this process actually works, just to get your feet wet. And then tomorrow, we're going to go into detail on these processes. So when you watch these last 2 videos here, what is DNA and how does it work, and what is a chromosome, it should be review. Hopefully it's review from grade 10. If not, it's just a broad overview of these processes to give you a general sense of what is going on in transcription and translation. And then tomorrow in the note, we're going to examine those in more detail. So that should be the last thing that you watch as part of Learning Block 2 here. And then Learning Block 3, I already mentioned this, but this is your opportunity to write the quiz, hand in your assignments, or complete any of your assignments from the day. Okay, I mentioned that there are new assignments for the genetic processes unit. You can see them over there on the left. So there is a genetic processes vocabulary list. So starting with the note and the little activity that you're going to do in review, there will be some vocabulary there for you to add to your vocabulary list. It's 30 words minimum, just like the last list, although you will likely have more. You should probably have more. And then the assignment is posted there. I'm not going to talk about the assignment in detail until we at least discuss what meiosis is, because it doesn't really make sense to go over the assignment because we just haven't even talked about that concept. So I'm asking you to do something and you have absolutely no clue what I'm talking about. So, or you probably don't. So I'll get into the details of that once we actually talk about meiosis together, but for now, just be aware that it is a sort of presentation that you're going to create, a video, that is going to show a biochemical process at work and explain that process in detail to me or potentially to another student, although another student's not going to watch it. But the idea is to communicate that information in a way that you would communicate it to another student. So it's pretty broad in terms of how you actually do that in the assignment. It doesn't have to be any particular method to show the information, but it does have to be a form of presentation or video. Do we start a new homework doc? Yes. So there is a new homework doc for every unit. So when you're starting today's homework, don't forget to create a brand new Google Doc that is genetics homework document, or whatever you want to call it, but genetics, and make sure the word genetics is in there somewhere. It is always a new homework document for every unit. So yes, you do not use the same one as you did for Unit 1. Okay, so those are the basic assignments. Don't forget that there's always a vocabulary list in the background. That's for the entire course. There will always be a vocabulary list that you can add vocabulary to for the particular unit that you're on. The iPhone camera went through the mitosis phases. Kim, I'm sorry, but I don't know what that means. Where do we submit our homework doc for this unit? Not yet. So at the end of this unit, you will see that there is a little post at the end of this unit that says Midterm Homework Questions Portfolio. At the end of this unit, which is pretty much the last day that you're with me before you go to your other course, I'm going to get you to hand in both of those homework documents for Unit 1 and Unit 2 in that assignment, and then submit it. Because I will be compiling your midterm reports then at that point. It's a little bit early in the course, but it's weird in the quad system. So anyway, that's there it is right there, Midterm Homework Questions Portfolio, where you would submit the first 2 documents. And then there's another one right at the very end of the course, which is not visible to you yet. In fact, I may not have even posted it up here yet. I got to add it. But there will be a second portfolio under evolution at the very end of the course where you'll hand in the rest of your homework documents. So I only assess them twice per quad. Do we hand in your homework doc? Not right now. So you will hand it in before you transition to your other course. It's one of the very last things that you do before you leave. So not today. At first, the iPhone camera only had one camera, then it split into 2. Mitotic division of cameras. Interesting. That's a similar idea, I suppose. Now I get what you mean, Cam. We are going to review mitosis, by the way, as well. So depending on when you took grade 10 science last year, you may have taken it during the weird time at the end, in which case I realized that not everybody completed every unit. It was a little bit hazy in terms of, are you supposed to complete it? Do you need to complete it? And some people just straight up did not complete it. You probably should have if you were planning on taking biology in grade 11, but at least the biology component. But we will do a review of mitosis for those of you that didn't get a real good look at it in grade 10. I mean, it's not as in-depth as we would have done in grade 10, but we do review it. Because mitosis is really important to know for meiosis, which we're going to talk about in this unit, because the processes are very similar. Okay, let me get right down to the note. Cells. Cell theory. Oops, a daisy. Why isn't that showing up? I don't know why it does that. That's a really unfortunate bug. All right, here we go. So you guys would have talked at least a little bit in grade 10 about the idea of cell theory. And that really, all that means is that all living things are made out of cells. Yes, all of them. And we don't count viruses. So definitely all living things, as far as we know, are made out of cells on Earth. So remember that a theory, or at least scientific theory, is backed by all the current evidence. It's an idea of how something works based on all the current evidence. And based on all the current evidence, all living things are made out of cells. Some are unicellular and some are multicellular. These are the tenets of cell theory, by the way, that you probably would have talked about in grade 10. The cell is the most basic structural and functional unit of life. You can't go subcellular and still be alive. If, for example, you were just to take a mitochondria out of a cell and just place it in really any environment, it would not be able to survive on its own. So if you go smaller than a cell in terms of subunits, you don't have a complete organism, a living thing. There's no way to do all of the functions. Now, mitochondria are really interesting in that they have their own DNA. And they likely, at some point in evolutionary history, were probably separate organisms that were then absorbed or entered another organism, and then those two organisms became symbiotically linked. And over time, the mitochondria lost its ability to reproduce on its own. But mitochondria have their own DNA. They do their own cell division within your cells. They do their own mitosis, really, mitochondrial mitosis within your cells. So they kind of are separate, except they can no longer survive on their own. So if you go below the cellular level, life cannot exist. What page am I on? Good question. I'm on the first page of the new notes package. So if you go to day 5, which is today, but underneath the genetic processes unit, and at the very first thing you see there is download this notes package, which is this notes package that we're currently using. So this is the notes package for the genetics unit. And I'm on the first page. You will notice there are 2 additional pages at the end of the diversity unit. Based on timing, I've cut those off. So I do occasionally talk about 2 additional concepts in diversity. It's the last 2 pages there, but we're not going to get to it, and it's not on any quiz, and so you don't need to worry about it. So I cross those pages out. Yeah, it's on page 21 of the whole notes package. So if you're using the one that I sent in the email, that's the complete notes package from beginning to end. I believe it is page 21. Yeah. Thanks, Michaela. Appreciate it. Okay, and then the last tenet of cell theory here is that cells arise, they come from pre-existing cells. They don't spontaneously generate, which is, it's an interesting idea. Obviously, based on observational evidence, we have never seen a cell spontaneously arise from its environment. However, evolution and the theory of evolution demands that at some point that was not accurate. So at some point there was some type of cellular spontaneous generation from non-living, potentially particles that we would call non-living. There was a first spontaneous generation of a cell. It happened at one point. However, in order for that to occur, like a lot of special conditions have to be met, environmental conditions, certain proteins and lipids and things like that must exist in the environment, and they can spontaneously generate as well, but this isn't the kind of thing that you're going to find in a glass of water. So very specific things are necessary in order for spontaneous cell generation, and we've never been able to recreate it. So I'm not going to pretend that like somewhere in a lab somebody has demonstrated a spontaneous cell generation with the right environment. It has never been done. So we make some educated guesses about how that process could happen, but we have never seen it happen. So it did happen at some point, or obviously it's highly suggestive that it occurred at one point, or that original cell had some type of extraterrestrial origin. It came from somewhere else, which I suppose is also possible. But there must have been some generation at some point. Anyway, but for our purposes anyway, there's no spontaneous cell generation. If you sterilize a container and completely remove all of the living cells from it, including prokaryotes and protists and whatever, as long as it's sealed, you're never going to see new living things arising within that container. And that's why canning food and things like that are a thing. You can kill all of the things living in a particular can, seal it, and then assuming that you've let none in, they will stay sterile. So your food will last for a long time, hopefully. We've added a couple extra things to cell theory over the years. One of them is the idea of energy flow within cells, which is that cells are able to take in, use energy, and produce waste products, all cells have some form of energy flow occurring. There's more to that, and if you ever take a really in-depth biochemistry course, they will examine that in far more detail than I am. But there is some form of energy flow within cells. Cells have hereditary information inside them. That is, hereditary means able to pass on to your offspring. And that information is DNA. So all living things use that same system, as far as we're aware. Any living thing we've ever seen uses DNA to pass on their information from one generation to the next. So every single living thing uses DNA for that purpose, which is kind of cool. It's conserved across all domains. And I mean, that obviously, again, is highly suggestive of common ancestry. And all cells have the same basic molecular composition. Lipid bilators are made out of lipids in every organism. Certain proteins are conserved across every organism. I already mentioned the DNA component, but the RNA component is the same across every organism. So there are a number of basic molecular compounds that are common across all living things. Surprisingly, we actually get into this in the animal systems unit in more detail when we talk about digestion. Because when we talk about the digestive system, we talk about how you get those materials into your body, those molecules that are necessary, that are preserved across all living things. And then at that point, I go into a little bit deeper examination of what those molecules actually are that you need to get in order to support your life. Anyway, so that's the basics. That's the basics of cell theory that you would have learned in grade 10, hopefully. But if not, those are some general, we'll call them rules, that apply to all living things. In terms of the structure of eukaryotic cells, and now I'm speaking specifically about eukaryotes here, keep in mind that all of the stuff that we're going to examine in genetics, almost everything, is going to apply to eukaryotes. Sometimes there are various versions depending on the eukaryote. So plant biochemistry and animal biochemistry are very similar. There are small differences that we are pretty much going to ignore for the purpose of this course. But eukaryotes in general have fairly similar biochemical processes, especially when it comes to how they read DNA and use DNA. And so for our purposes, all of this stuff is going to apply to eukaryotes. Prokaryotes have different systems to do these things. They read DNA and make proteins in a similar way, but they lack most of the structures, the organelles, that assist in that process that eukaryotes have. And so they actually do it fairly differently. And so I'm not going to pretend that this information applies to prokaryotes very well. It doesn't. If you're interested in how these processes work in prokaryotes, take a molecular biochemistry course on microbes in university. You may need to get into a biology degree in order to do that, but I highly recommend it. Or actually, you can find videos on YouTube if you just have a personal interest. But if you're interested in going that way in your career, you're fascinated about the biochemical processes of prokaryotes, there's lots out there. Obviously, you can spend a whole career investigating those things, but I'm not going to dig into it too much in this course because it's just not relevant for what we're going to talk about. So this part right here is actually something I'm going to let you do on your own. So it is a reading about what the organelles do, and there are some instructions here to color them. You don't have to color them. This is totally up to you. I don't know why I'm using yellow to color in the brown, but that's probably triggered some people in the audience. If you want to color these, you can. As a memory tool, coloring is helpful for some people. When I took anatomy, my first year anatomy course, a human anatomy, when we just learned, like, these are the different muscles of the arm, these are the bones of the wrist, or whatever. When you learn those things in an anatomy course, a human anatomy course, one of the required texts, I didn't buy it, but you could buy it, is an anatomy coloring book where you go through and color them, you color code them to use as a memorization tool. And many of my friends told me that was how they learned it. it was very helpful for them to go through and color and be like, this is a nucleus. And you color it in purple. Purple is the nucleus. You go back and look at it again, you're like, yeah, purple, nucleus, or whatever. That seems to be a tool that's valuable for some people. It doesn't work for me. I've tried it. And I don't know, maybe I don't think about it enough while I'm doing the coloring, but I get nothing out of it. So I'll leave the coloring up to you. If you want to color this, it's a little hard to do in Cami. You're welcome to color the cells. But if you don't want to color them, you don't have to. I'll leave it up to you in terms of whether you think that that's beneficial for you in terms of a study aid. But anyway, it goes through, it describes each of the organelles. And then as you do it, what you're supposed to do is think about that function. And while you're thinking about it, go to the cell, find that particular organelle, and color it, thinking about what it does. Oh, this is the thing that makes the energy molecules for the cell. Oh, this is the thing that folds completed proteins and packages them for export. This is the thing that contains the DNA, and while you color it. Okay, so that is a learning tool that you could employ if you'd like, but I personally would just label this and then look at the labels and then think about it. I don't require the coloring component. I'll let you decide. So you're going to also label this as you go. I've posted this in the notes folder. If you're looking for a completed version of this, it's in the notes folder. But you probably can label it based on what you know from last year and based on the descriptions. But if not, again, the answers are posted there for you. If you're really not sure, or you're looking at it, you're like, I don't think that's correct, let me know. Now, I will say that I believe that on your copy I added a vacuole. I'm not sure if I actually added it to the blank one or not. This looks like I added it just to my note here, but I believe there is a vacuole. And I just drew one in because this diagram does not contain a vacuole. So I'm not trying to confuse anybody on that, but there should be a vacuole there, which looks like nothing. I mean, it just looks like a circle. It's an empty sac. It's not empty. It contains mostly water, and water soluble nutrients. But it doesn't look like anything under the microscope. It looks like an empty sac. So anyway, and here are the questions that you're going to do on your own after that. There is a vacuole? Okay, good. I remember to put it in there. I'm glad. I wasn't sure. So these questions are, again, based on the reading that you just did for the cell. So I'm going to leave that for you to do in a minute when I'm done the note component here. Okay, so I want to talk a little bit about DNA and the structure of DNA. Tomorrow we're going to get into what DNA is for and how it works. But today I just want to talk about what it is. Okay, just on its own. Because the term gets tossed around all the time. And I would say the vast majority of people don't even know what it means. Like, what is DNA an acronym? Or is it DNA? Is it a word? It's an acronym. It stands for deoxyribonucleic acid. And the name deoxyribonucleic acid is descriptive of the actual molecule itself. So we're going to look at the molecule in a second, but I'll show you where the deoxy is and where the ribose is, so that you can see why it's actually called that. But anyway, that's the name. Deoxyribonucleic acid is the molecule that contains all of your hereditary information. It's a giant macromolecule. And it is the starting point for virtually every important cellular process. It's how you regulate everything that's happening in the cell and build everything that's happening in the cell. And you regulate it by building it. So if you want the cell to do more of a particular thing, then you build more of a particular structure, or build more of a specific chemical messenger, or build more of a whatever. So most of the regulation comes from turning on and off specific genes, saying build more this, build less this, which is also a function of the DNA. So the DNA can also turn things on and off. So the main function, what DNA is primarily doing, is it is an instruction set for making proteins. And like I said, we're going to get into that process in great detail tomorrow. But why proteins? Well, proteins are necessary for building, maintaining, and replacing all of the tissues of your body. You may be wondering, you're looking at your skin, and then you look at somebody's eye. You're like, whoa, those are different. They're really different. You can put them under the microscope and look at the various tissues under the microscope, and they are structurally different. They are biochemically different, but they have the same DNA in them, which is weird. The same instruction set made two completely different things. And the key there is that those cells have different parts of their DNA that is being read and turned into proteins. So a cornea cell that would be in the clear coat in front of your eye is reading a specific part of the DNA and making specific proteins that allow it to be clear and have all the properties of a cornea cell. And a skin cell is reading a different part of the DNA and making different proteins associated with being a skin cell. So it's all about which proteins are different. That's what makes your different tissues different from each other, is the proteins that are made, because they have the same instruction booklet. They're just reading different pages from it. So aside from that structural role, proteins are also classified as enzymes. Some are classified as enzymes. An enzyme is if you've taken chemistry already, it is a form of why this word is on the tip of my tongue, now I'm just going to forget it. It modifies the rate of a chemical reaction without participating in it. That's the definition. It is called a catalyst. There you go. I had to think about that for a sec. It's a catalyst. So an enzyme is a catalyst. It allows or changes the rate of a chemical reaction happening in the body without actually changing itself. So you make a whole bunch of different kinds of enzymes that are all the different chemical processes inside of your cell are modified by enzymes. Your digestion is modified by enzymes. You may have heard of things like lactase or protease, or these are digestive enzymes. Thank you. Thanks, Anastasia. Catalyst. It's too bad there's such a delay here. Anyway. So that is one role of proteins. They can be enzymes, and they can also function as hormones. Hormones are signaling molecules. If one cell in the body wants to tell another group of cells or another cell, stop doing this or start doing this or change this about yourself, hormones are required. So hormones are signaling molecules. For example, when you start to go through puberty, the pituitary gland in your brain starts producing follicle-stimulating hormone, which in turn triggers the production of testosterone and estrogen, which are also hormones. And those hormones are going to interact with receptors on cells all over your body. And those are the things that cause your secondary sexual characteristics to appear. Body hair, etc. Everything associated with puberty. Growth as well. It triggers the production of human growth hormone, HGH, which increases your growth as a human. Again, interacting with a bunch of different cells in your bone plates and your growth plates in your bones and stuff like that to increase your growth. So that's all a system of hormone communication that's going on within your body, which are just proteins. There's protein signalers. What delays or changes hormone production? That's a very general question, Cam. Do you mean sex hormones? We're talking about like the ones that are upregulated during puberty? Because that's a really broad question. There's many different types of hormones, thousands. And they all have different purposes for signaling in your body. There's hormones that signal hunger, and hormones that signal satiation when you're no longer hungry, and hormones that tell you that it's time for you to go to the bathroom, and things like that. There's all kinds of hormone communication going on. Although I should say that the bathroom thing is also a muscle stretch response through your spinal cord. But there are hormone communications that are associated with a lot of these. For sex hormones, what triggers the pituitary gland to begin releasing follicle-stimulating hormone and begins that cascade in puberty? That is not my area of expertise. I don't know the answer to that question. I don't know. I don't know what turns on FSH. That would have to be that. I'm sure that's known. I'm sure that's known because I'm pretty sure that that people that have illnesses that are associated with delayed onset puberty, that where puberty is not occurring, I think you can take things to either activate FSH production or to simulate FSH production. I'm sorry. I don't know the answer to that question. I'd have to look that up. If that is in fact your question. Anyway, so those are the main things that proteins are doing, and all of that, the instructions for building all of those things are found in your DNA. Okay, so that's why DNA is important. It makes all of the important functional units that are doing things inside of your body. So to reiterate here, DNA is a genetic or hereditary. Hereditary means you're able to pass it on to your offspring material. It is in fact the only hereditary material. It's the only thing that you get from your parents that is different from person to person. Everything else that you're getting from your mom or your dad, mitochondria, Golgi apparatus, those things are all identical. But the genetic information is different. And I mentioned that because we only found that out within the last 100 years, fairly recently, that DNA was responsible for that hereditary passing on of information from parent to child. It took us a long time to figure that out, actually. And it's found in the nucleus. You guys already know this. I say of most of all cells, but I had to put a little caveat in there and say mostly all cells, because some cells actually remove their nucleus when they become mature. So a perfect example is red blood cells. Once you create a mature red blood cell, the nucleus is removed. It's enucleated. And so it doesn't have the ability to produce new proteins once a red blood cell is finished, which means it has a very prescribed life cycle. After a certain time, it exhausts the proteins inside of it that are being used to maintain the red blood cell, and then it needs to be removed from your bloodstream via your spleen. So not every cell has a nucleus in it, but almost every cell. In that cell, in any cell, you have 46 big long segments of DNA, pieces of DNA, sticks, if you will, of DNA. And those are called your chromosomes. Okay? And when I say you have 46, I mean you're a human, so you have 46. Not every organism has 46. So it differs dramatically between organisms. Some organisms, which you may actually use as part of your research project at the end of this unit, only have 6 or 8 chromosomes. And these are eukaryotes. Prokaryotes, again, none of this stuff applies to prokaryotes, but prokaryotes usually just have one single loop of DNA, as I mentioned before. They're not found as chromosomes. So, but if you unravel them, so if you take one of those sticks and you kind of stretch it out, which you can't really do in real life, but if you could theoretically do that, you would see that the ladder structure of DNA is what's called a double helix. So a double helix is a ladder, and then both of the ends of the ladder are twisted in opposite directions. So if you were to twist this end of the ladder this way and twist this end of the ladder this way, you would create a double helix, which I'm not going to do a good job drawing this, because this would be one end of the helix, and then the other end would be like this. I got a better picture of it below. I'm sure I've got a double helix below. Yeah, there she is. Beauty. So there's a double helix structure. Yes, you can. Yes, you can. I wouldn't include things like the organelles from grade 10, because, I mean, that's rudimentary cellular biology. That you probably learned those in grade 8, actually. But But some of this DNA terminology, I would say, yeah, that's definitely core terminology for genetics, and I would say that's totally fine to add to your vocab list. Okay, so anyway, yeah, you got this structure here, also known as a helical ladder, and it's comprised of alternating sugar and phosphate molecules. Like I know you know what a sugar is. You eat sugar. But maybe on a molecular level, you're not aware that it has like this pentagon shape, where there would be carbons at every corner of the pentagon. The actual molecular structure of sugar is not something I'm ever going to ask you about, but if you take grade 12 bio, you get much, much deeper into the biochemistry of these things, and you definitely need to know the structure of sugar. So I won't ask you in this course, but just as a heads up, eventually you're going to need to know the structure of sugar. So it is a pentose sugar. It has 5 carbons. And a phosphate, hopefully, phosphate sounds familiar to you from grade 10 chemistry. Phosphate is PO43-, and that's no different. Phosphate is phosphate. And so there is a sugar, and then a phosphate, and then a sugar, and then a phosphate, and they're linked together. So getting back to the backbone here. So if I were to unwind this helix on the left, it would look like this. Sorry, I've made some annotations on here. But if you look at just the backbone component, which is right here, you'll notice that it is an alternation of a phosphate, then a sugar, then a phosphate, then a sugar, then a phosphate, then a sugar. So we call that the sugar-phosphate backbone. And there's one on both sides. I've also mentioned, I've drawn on here that there are 2 ends of the DNA. There's what's called the 5' end and a 3' end. And on the other side, it runs in the opposite direction, the 5' end to the 3' end. What the 5' and the 3' mean is which carbon on the sugar, of the different carbons on the sugar, which one is bound, or sorry, which one is open, okay? So the 3' sugar is open on this end, and then on the other end here, the 5' sugar is open. But that is not something that you need to know in grade 11. So this comes up in grade 12 quite a bit, but not in grade 11. The reason I write it on here is because it's just important to know that there is a direction that exists in DNA. And when you read the base pairs, so the base pairs are in the middle. There are these letters in the middle right here. Okay, and when you read them, you only read one side, and you always read them in the same direction. And what that direction is not important for grade 11, but it is important to know that you read them in a particular direction. You read along in a certain direction, and you don't ever read it the other way. Because obviously if you read the DNA, the instruction book, backwards, it would be meaningless. It doesn't say anything. If you read a book in reverse, it doesn't say anything. So, and the same thing is true for DNA. So there is a directionality to it, and it's always read in the same direction. So, like I said, we've got this sugar-phosphate backbone where it goes sugar, phosphate, sugar, phosphate. That sugar, by the way, is called deoxyribose. And that's where the deoxyribonucleic acid comes from. But the reason it's called deoxyribose is that the type of sugar is a ribose sugar. That is the name of a sugar in that configuration, with those hydrogens and oxygens bound in those particular locations of the sugar. All sugars are the same shape, that pentagon shape, but this one has certain oxygens bound in certain places and certain hydrogens bound in certain places, which we, again, don't need to know for this course, but that's why it's called deoxyribose. There is a particular location on there that does not have an oxygen, and that's why it's called deoxyribose, the D meaning missing an oxygen at that location. The other type of ribose sugar is called just ribose. It's not called deoxyribose. And that's what RNA is made out of. So RNA is a lot like DNA. It has a sugar and a phosphate and a sugar and a phosphate down the backbone. But instead of having deoxyribose for every sugar, it just has ribose for every sugar. But you'll notice that the actual formula here for this deoxyribose is the same as glucose, which is just like sugar, the table sugar, right? C6H12O6. It's a sugar, so it does have the same chemical composition. It just has a different arrangement of the hydrogens and oxygens. Okay? When we talk about a nucleotide, now here is a great term to place into your vocabulary list. A nucleotide is one unit of DNA. And when I say one unit, actually, let me pick a good color to highlight this. Maybe use blue. I don't have blue in here. It is a sugar, a phosphate, and the letter that you find in the center, which is known as a nitrogenous base. Okay? So we have one sugar, one phosphate, and a nitrogenous base. That is like the base unit that DNA is made out of, because it's that repeating over and over and over again. So that subunit, a phosphate, a sugar, and a nitrogenous base, is called a nucleotide. If you string a whole bunch of nucleotides together, and then you string a whole bunch of nucleotides together on the other side, and then you bond them together, you get DNA. That's one nucleotide. Phosphate, a sugar, and a nitrogenous base. So, you may be wondering then, what's up with these nitrogenous bases in the middle here? Well, those are the actual letters of the code, because the sugar phosphate just repeats and repeats and repeats. There's no information in that. You can't know anything from reading the backbone of the DNA. What you want to read are these guys in the middle. Okay, and as I mentioned, you're going to read them along the sugar phosphate backbone. So you'd be reading them in this direction right here. Okay, so for example, this would be A, G, C, T. That would be like the code that is shown here on this strand of DNA. Okay, and you only read one side. Which side you read, we'll talk more about that later. But it's only one of the sides that's read. The other side is called the complementary strand, which is really useful. So if you look at which letters here of nitrogenous base are attached together, the letters represent the different types of nitrogenous bases. There are 4. You can see all 4 here. I also mentioned them up in the note up here, so I'll go back up to that right there. So the 4 rungs of the ladder that you can get are adenine, guanine, cytosine, and thymine. You must know these. You will never take a biochemistry course where you don't need to know that. That's like the most elementary thing that you need to know about genetics, is that there are 4 possible letters that you can make a DNA rung out of, and they are adenine, guanine, cytosine, and thymine. And you'll notice that I've connected 2 of them together here with an arrow. And that's because those 2 always are paired together across the center of the ladder. If you have an adenine on one side, you must have a thymine bound to the other side. You have to. They physically are chemically incompatible with each other. Their bonds will not allow them to sit adjacent to each other because of where those chemical bonds sit. They will be pushed away, and that you will not be able to bond that DNA together. So you have to put a thymine across from an adenine. And the other combination that you always see is guanine and cytosine. So guanine must always be paired with a cytosine. So you need to know, like I said, the 4 base pairs, adenine, guanine, cytosine, and thymine. And you also need to know which 2 bind together, which pairs always exist. So there are various methods that people use to remember this. Usually it's some trick where you create an acronym that goes AT, because A always goes with T, and CG. The one that my teacher had when I was in high school was all teachers give candy. That was what was my grade 11 biology teacher's name? Oh, I can't believe I forgot it. I love that class. Shoot, I'm actually kind of upset that I forgot her name. She was a lovely teacher. She's one of the reasons why I wanted to go into teaching. But anyway, all teachers give candy, or whatever you would like to do. I mean, obviously you can rotate the G and the C there, because as long as they're together. Anything you like. You just create a mnemonic device, something that will help you remember A and T goes together and C and G goes together, because we're going to be using that skill to decode the DNA a little bit later, tomorrow actually. But if you go down to the diagram down here, you'll notice that A is always across from T and C is always across from G. Okay, so those are called complementary base pairs because they are the ones that fit together. They complement each other. The really cool thing about this is because there is always one complementary one that will fit across, it is a strategy to protect your DNA against accidentally messing it up. For example, if you go out in the sun, you're out on the beach and you don't wear sunblock, don't do this, and a little bit of UV comes and blasts one of your pieces of DNA, okay, in one of your skin cells, which, by the way, happens all the time. And you, unfortunately, UVB and UVC, which is what you get from the sun, are ionizing radiation, which means that they're strong enough to break chemical bonds. So it might take this cytosine and blast it out of your DNA. Okay, so you got a gap there, and this does really happen all the time. One of the really cool things about this is you have enzymes that will run along the DNA and look to make sure that there's no holes. And when they find a hole, they will attempt to insert the base pair, the correct nitrogenous base that fits in the missing hole. And the only thing that fits in that hole is cytosine. So that protein will zip along and it will add a cytosine back into the missing hole. Cool. It's an automatic repair mechanism. And the same thing is true if you were to accidentally blast out the guanine. Well, the protein could come down and say, okay, well, you got a cytosine on the other side, so that must mean that this is a guanine. And so it does provide a little bit of a protective effect to have this ladder configuration. It's basically like a backup copy that is attached to the other side of your DNA. Okay, it's error protection is what it is. And that is so useful because remember, this is the most important molecule in every one of your cells. It tells your cell how to do everything. So if you mess this thing up, your cell goes haywire or it has to commit cell suicide, apoptosis, because it just is no longer functional because you've messed up the code. So you really do have to protect the code. And so this configuration, this double helix, is extremely protective. Okay, that's all I'm going to say about the structure of DNA. Are there any questions about this? Anything that I didn't explain clearly? Regarding, like, what does DNA actually look like and what's its composition? Okay, I'll take that as a no. If anything comes up, let me know. I realize there's quite a bit of a delay. Okay, a couple other little things about DNA. Okay, I've talked about the basic structure and how it's put together. Your DNA... Oh, actually, this definition is probably one of the most important definitions that you learn in the entire course. And that is that there are sections of your DNA, specific lengths of your chromosomes, that make a protein. One protein will be like this area right here is a protein, and this area here does one protein. That area that does a single protein is called a gene. So when we're talking about genes and genetics, We're talking about the sections of your DNA that code to make one single protein. So this area makes a protein, that's a gene. This section makes a protein, that's a different gene. Okay? So a gene is a section of DNA that makes a protein. If you were to go through and read your entire genome from beginning to end, and remember that you just read along the one side of the ladder, A, G, C, T, 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 G, 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 C, T, 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 G, T, 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 C, whatever. You can read the whole thing from beginning to end. It is approximately 3 billion base pairs long. So it's pretty long. You got a heck of a long code there. And remember that every time you make a copy of your cells in mitosis, you have to make a copy of the entire thing. A perfect 3 billion base pair copy. It's not perfect, by the way. You make errors every single time, but you try and make it as error-free as possible. Considering how long it is and how often you do the copying, we have a remarkable ability to have low errors. But it's long. Okay, 3 billion base pairs. We actually did that, read an entire human genome for the first time. I guess it would be when you were born. Yeah, about 16-ish years ago now. Which is crazy. I mean, it's not that long ago, guys. I was in high school, and we for the very first time read the entire genome of a human. That was when they finished the Human Genome Project. Like, that's insane. That's really not that long ago. Interesting question, Trenton. This is a little bit outside the purview. Fair enough. I'm actually not a baby boomer, by the way. I'm a millennial. But people make that mistake all the time. The fetal alcohol syndrome is not a genetic disorder. So your genes are not affected by alcohol consumption. It's the expression of the genes that is affected by the alcohol consumption. So it's not the DNA itself that is altered by alcohol consumption. It is how you use the DNA to make proteins. That process is disrupted through the consumption of alcohol. And generally, the problem with FAS is really that you have so much cell division and cell replication happening during embryological development that if you mess with that process at all, inhibit it even in a small way, it inhibits how the DNA is read and how it is made into proteins. Yes. Even if you mess with the reading and writing of DNA and creation of proteins, even in a small way during embryological development, it can have profound consequences because there's so much cell division actually happening. So even if you're only inhibiting it for a small period of time, the down the line effects of that are so significant. And usually FAS is not a single incident of alcohol consumption. It's sustained alcohol consumption for an extended period. And yes, it does have negative long-term side effects for developing fetus. So, but it's not genetic specifically. It's more in gene expression. It's how the genes are used. So I hopefully answered your question. So then a great question would be, how many genes do we have? How many sections that make proteins? About 30,000. So humans have about 30,000. This is obviously different per species. If you look like at a groundworm, a regular earthworm, a North American earthworm, which is an annelid, they have about 18,000. So that's a typical number for a complex eukaryotic organism. We only know the function for about, this is a couple years old, 50% is a couple years old. I say it's probably closer to 60% now. We only know the function for about 60%. And when I say that we know the function, I mean that we have a general sense as to what it does. When there's more gene expression from this gene, we see this in humans. When there's less expression, we see this. We don't know exactly what it does, but we have a general idea that it is involved in this kind of process or makes this kind of protein, not necessarily exactly what it makes or exactly what it does. So it's a little bit of a misnomer to say that we know the exact function for 50%. But keep in mind that the other 50, or I would say it's closer to 40 now, the other 40%, we have no idea what it does. So there's lots of genes that it's just like, well, okay, well, that's a section of your genome, but I don't know. It's like a black hole. We have no idea what that does. So I think that's pretty good that we know that much about what those specific genes do. Certainly, we've discovered all of that stuff in the last, I don't know, not a long time, your lifetime. We've discovered all of that. So I think that's pretty good. About 1.5% of that 3 billion base pairs, okay, actually encodes for proteins. So those are the sections that we would call genes. Only 1.5% is genes. So that might seem weird to you. You're like, well, what's all the rest? Like, most of all this other DNA, there's like so much other DNA that isn't genes. Well, we used to call it junk DNA. I'm going to put a line through that because we absolutely do not call it that anymore. We used to call it junk DNA because we thought it did nothing. But it does so many things. So what it actually does is it is the on-off switch for the genes, for the other genes. That section of the DNA is able to turn on and turn off all of the other sections that code for things. And that's a complex process. What is turned on in an eye cell and turned off in a skin cell? It's pretty complex in terms of how to regulate that, turning on specific things more, turning them on less. And so the rest of the domains of your DNA, the areas of your DNA, are all involved in that process of turning things on and turning things off. So now, and that you can, this is definitely a great term to put in your vocabulary list, but that section, those sections are called introns, I-N-T-R-O-N-S, introns, or non-coding DNA. Typically the word intron is used though. So introns are really important for regulating what is being used, which parts of the DNA are being used. They are not junk. Junk is a terrible term. They used to call it junk DNA. That is a terrible term for that. Because it couldn't be farther from the truth. Why aren't there fetal alcohol syndromes for other drugs? There are. They don't seem to be quite as profound. I mean, obviously I'm making a extremely broad generalization here, but you're talking about two opiates here, cocaine and well, actually, meth is not an opiate; it's a stimulant. But those absolutely do affect fetal development. The difference between that and alcohol consumption, and sorry, this is a bit of an aside, guys. We're going on a little side journey here, but is that they just simply aren't as common. Fetal alcohol syndrome is far, far more common than those other ones. And the symptoms that you get from fetal alcohol syndrome are fairly consistent among children who have been exposed during fetal development. So because it's a common symptomology, and it's very common to have alcohol exposure during fetal development, that there is a clear syndrome that is involved with alcohol. It's not as cut and dry with methamphetamines and cocaine and things like that. There are effects on the fetus. With opiates, they're not as significant. I don't know that much about meth specifically, but it's not as clear cut. And often those drugs are used with other drugs, and so there's like a milieu of different chemical compounds that are involved, and it's not as clear cut as fetal alcohol syndrome, which is typically just alcohol consumption, although it can be found with consumption of other drugs as well, but it's just not as likely. That's right. A syndrome is a set of symptoms. That's what a syndrome is. A syndrome is simply just a set of symptoms that tend to occur from a particular circumstance. It's not a disease. A syndrome is really just a set of symptoms. Technically, something other than alcohol could cause fetal alcohol syndrome, the same set of symptoms. But typically it is alcohol. So that is what a syndrome is. Okay, anyway, how do we organize them in terms of number? Well, chromosome 1 is the biggest one, and chromosome 21 and 22 are the smallest. I think actually 22 is physically smaller than 21, chromosome 21, but it has slightly more genes than chromosome 21. Okay, so when we call something chromosome 1, what we just mean is the biggest one. The biggest one is 1. The smallest one is 22. 22 is physically the smallest. It looks the smallest. And 23 is the sex chromosome. You'll notice that I've only named 23, and that's because each of those 23 has a homologue. Oh, bringing up terminology from the diversity unit. A homologue is a piece of DNA that does the same thing, but is not identical. So each of your chromosomes has a homologue. So in total, there's 46, but there's only 23 and their homologues. Okay, you could call them pairs. They are called pairs. They're called homologous pairs because there is a homologue to it. Okay, so if you were to look at this, like lay it all out on a piece of paper, which we used to do, during embryological development, you can take a sample of the amniotic fluid. You can lock a cell into a particular phase. I think it's metaphase, if I remember correctly. You guys remember metaphase, hopefully, from last year. And then you can lyse the cell, burst the cell under a microscope, spread the chromosomes around, put a specific dye on them to make them more visible, and then you just take a picture. And then once you take a picture, in the old days, they used to develop the photo and then cut them out with scissors and then arrange them by hand on a piece of paper. But now, I mean, that's obviously way too much work. Now we do like computers do it. So computers take the picture, they do the organization. It's all automated. So much handier. And then you get a picture at the end of your chromosomes. And that picture is called a karyotype. So this is a human karyotype. It is a picture of your chromosomes, okay, of someone's chromosomes. So you'll notice here that chromosome 1 is the biggest one. You'll also notice that there are 2 chromosome 1s. And the reason that there's 2 is because you have homologues. They're not identical, but they have the exact same genes on them. So you have one from your mom and one from your dad for each chromosome. And those are called maternal and paternal chromatids. I probably should write that on here somewhere. So the ones from mom are called maternal, and the ones from dad are called paternal. Okay. What I wrote up there in red there is just what I just said about you take a sample from the amniotic fluid. And you can use some chemicals to freeze the cells in metaphase, break them open, and take a picture. So here are all the homologous pairs for humans, and you'll notice that the very smallest one here at the end is number 22, although technically it has slightly more genes than 21. And then the last one are the sex chromosomes. And this is going to be different depending on if you're a male or a female. If you are male, you're going to have an X chromosome at the end and a Y chromosome. The Y chromosome it's not shown in this picture, but it's like a little tiny guy. It's just like a little funny shaped one. That's the Y chromosome. And if you're female, like this individual is here from this photo, you're going to have 2 X's. So this is the sex determining chromosomes. And they've been organized again here first by size. So they arrange them from largest to smallest. Then if they're the same size, and it can be difficult to see their size especially when you get to really small ones, and 16 and 17 are relatively similar in size. Then you look at the banding pattern. And then if the banding pattern is very similar, then you look at the centromere location. So the centromere is the little part right here on each of these. There it is here. Here's the centromere here. It's where the chromosomes are attached when you replicate them. So if I were to make a copy of this chromosome right here, which you do, when you make a new cell, right? You're going to make copies of these. The copy is going to be attached at the centromere, and then it makes like that X shape that you guys probably saw during mitosis last year when you looked at chromosomes. So they are attached to their identical sister, those are called sister chromatids, at the centromere location. So that's the centromere location is also used to help organize here. Sorry, there's some questions in the chat. I read that people can be other genders. Yes. So there is a common confusion among people that gender and sex are the same. They're not. Those terms mean different things. We're often, at least in North American or Western context, we seem to be very comfortable believing that they are the same term. They are not the same term. You can't change the meaning of those words. Sex is a biological thing. It's what parts do you have when you are born. And it is determined by chromosomes. That is unassailable. That is how it works. Although I will say that there are some chromosome anomalies that you can have where the sex doesn't necessarily match up with the chromosome assignment. So it's not a perfect system, but in general, the chromosomes determine sex. But gender is a societal, cultural construct. What do we consider maleness to be in society? What do we consider femaleness to be in society? That's gender. That's a construct of culture. It's a cultural construct. And we can decide whatever we want gender to be. That is decided culturally. So it has nothing to do. There is no biological reference for that. I mean, if we culturally decide that it is biologically linked, then it is. And if we culturally decide that it isn't, then it's not. It is culturally determined. So those are not the same. Okay, sorry. Going back to sex. So you mentioned the sex part. Can people be born other sexes other than male and female? Kind of. You can be born with both. So it is possible to be born hermaphroditic with both male and female parts. It is also possible to have dual sets of male or female genitalia. And these are usually based on genetic anomalies, but sometimes they're environmental and have to do with exposure during fetal development, but not always. Not always. These are extremely rare. We're talking about very rare circumstances here, but it is possible for those situations to occur, and they do occur. And they're dealt with medically sometimes, and sometimes they're not. So that's sort of like a tricky area in medicine. How do you deal with that? I don't have a good answer for you on that one. But does that exist? Yes, it does. And it doesn't necessarily always correspond to the genetics, although it often does. Sorry, I've had a crash here. Why isn't this working? Come on. Every once in a while, this thing wants to keep me on my toes and just says, no, I'm going to stop working now. Come on. I wish there was a way to do this with a cord, like a direct connection that wasn't over Wi-Fi, because the Wi-Fi is so shady. Like it wants to work 1 minute, and then the next minute it's terrible. This isn't the same time of day that this happened yesterday, is it? Could this be time of day related? Jeez. I was almost done. I'm right at the end of the lesson here. This is killing me. Come on. Work. So frustrating. Like, it says I have an excellent connection to the network, so why is it? I wonder if I've got to restart this air server. Darn, I was so close to being finished. That drives me nuts. It worked so well all day. What a pain. Sorry, guys. All of a sudden, this has decided to stop working. That is infuriating. Oh. Oh, okay. Okay. All right. Okay. This is the end of the note. Okay, so this is, again, just basic facts here about DNA. There's 23 pairs of chromosomes. Okay. I mentioned this in the other lesson, but we call that number the half number of all of your chromosomes, the haploid number. So haploid is, my pencil's going to stop working. The haploid number, come on, is half of the full number of chromosomes. And the reason why we even care about that number is because the other half are just homologs to those first half, to the first half. So that's actually an important number to know, which is half of the total number. It's also sometimes referred to as the 1n number, or the n number. We arrange the chromosomes in order of size, from largest to smallest. There are 46 total. So if you were to actually go through and count up all the sticks, there's 46 sticks, 46 lengths of genetic material. And then that full number, all of the lengths put together, is called the diploid number, or the 2n number. You have 22 pairs of autosomes. Autosomes are the chromosomes that are not related to sex determination. So there's 22 of those. And then you have 2, the last pair, which are called allosomes. Again, great terminology to add to your list here. But allosomes are the sex-determining chromosomes. They're usually written at the end of the karyogram. In the image above, it's a female. And the reason you know that is because there are 2 X chromosomes. If it was a male, there'd be 1 X and 1 Y. And then I have a little image over on the side of what that would look like. So there would be 1 X, and then the Y is just a tiny little guy. Yes, short answer to your question, Lillian. A person with Down syndrome has 3 of chromosome 18, trisomy, or is it 21? Trisomy 21? No, 21. It's 21. Trisomy 21 is, or is it trisomy 18? Now I got to look this up now. It's one or the other. 3 of one of those, and so they do have 47 in total. That's correct. It's not 24 pairs. It's 23 pairs plus 1. So the last one is not a pair. It is just an extra. And we're actually going to talk about some other genetic illnesses similar to Down syndrome that arise from problems when you create your sperm or egg. Those are called non-disjunction events. And we're going to talk about those in quite a bit more detail when we talk about meiosis and how you make sperm and eggs. It's actually one of the main parts of the genetics unit in this course. Okay, I'm going to stop talking now. Sorry, I'm mad that I had that technical problem and I wasted some time. Sorry, guys. I don't like wasting your time. What you're going to be working on right now is there's a quick review video, a tour of the cell. You're going to read the section on what the different organelles do. You're going to label a diagram of the cell. There is a completed version of this, like I said, in the course notes if you want to look into it, and to compare. And you're going to answer the questions on page 3 of the notes, which again are just questions about the functioning of these. If you're having any trouble with them, again, drop by the Google Meet in Learning Block 3, or you can ask me at the beginning of class tomorrow. I should always have a little question period at the beginning where you can ask questions about the homework. The second part here is a quick little summary. These videos are not long, they're 5 minutes each, of the process of taking DNA and turning it into a protein. That is called protein synthesis or transcription and translation. We're going to get into this in great detail tomorrow, but it's good before we head into that to have a basic explanation, to watch a basic explanation of it. That way we can jump right in tomorrow. So that's what these 2 videos are at the bottom. Again, as usual, add anything to your vocabulary list, but do not forget that today is the last day of the diversity unit and you need to write a quiz. So if you feel ready to do that, I highly recommend doing it during Learning Block 3, and the reason being, if you have any trouble while you're writing it, that you can just drop into the Google Meet and ask questions. It's the easiest way. Okay, guys, I'm going to let you get to work, and I'm going to stop talking. Apologies for my technical issues.